Proteins are big molecules. They are polymers. So polymers are big molecules, large molecules. They are made up of small units. And these building blocks of polymers are called monomers. So proteins are polymers, and the monomers, the basic units that make up proteins, are amino acids. And you can see the example here, the horn of the animals are proteins. So proteins have many functions. So here we are going to classify proteins by their functions, give the names and abbreviations for the amino acids that build or make up the protein, and draw the structures of these amino acids. So that is the goal for us right now. So functions of proteins. Like I said, the proteins, we are going to classify the proteins based on the functions. So the proteins are polymers. They are made up from 20 different amino acids. So like we said, the amino acids are the building blocks, the monomers. They differ in characteristics and functions, and depend on the order of amino acids that make up the protein. So what we are saying right now is the amino acids are the building blocks of proteins, and the order of the amino acids in the protein determines the function and the characteristic of that protein. So the way these individual amino acids are connected, the sequence is important. From structural components such as cartilage, muscles, hair, and nails, they function also as enzymes to regulate biological reactions, digestion, metabolism, cellular metabolism. So there they function as enzymes. In the structural situations, they function as cartilage, muscle, hair, they build all that. And in transporting of oxygen in blood through hemoglobin is also a protein. Hemoglobin is a protein. So this is helping in the transportation of oxygen in the blood. So proteins have various functions. So there is a table. This is in your book as well. So please refer to your book. If you are unable to see this clearly, refer to the book. And you can see the different classes of proteins based on the function and where they are located. So when we are looking at proteins for protection, we are talking about immunoglobulins. This is the final, here I'm looking, that stimulate the immune response. So that is one of the functions of proteins. They are present as immunoglobulins. When it comes to enzyme action to catalyze biochemical reactions in the cells, trypsin is a protein also, and sucrase, hydrolysis of sucrose. So these are again enzymes which are proteins that help in the biochemical reactions. Then as hormones that regulate body metabolism and the nervous system, you have insulin, a very important hormone that regulates glucose levels, is a protein. Then as storage, to store nutrients, these are your proteins that store nutrients. And for transport, they carry essential substances throughout the body. Hemoglobin and lipoproteins that we discussed in the previous chapter, lipids that transport lipids are proteins. So they help in transport as well. They make muscles move. So these are the proteins, myosin and actin. They help contract the muscles to move the muscles. And there are structural proteins, like we talked about the horns, keratin in hair, skin, wool, nails, collagen in tendons and cartilages. So we see proteins at various locations doing various functions, and all these characteristics and functions are based on the sequence of the amino acids that build up the protein. Now we are looking at the monomer, or the individual molecule that makes up the protein, the amino acid. So like I said, amino acids are the molecular building blocks of proteins. So this is a structure of an amino acid, we can see that it has many components to it. And this is the ball and stick model. So this is also a complex molecule, you can see that, and it has a central carbon. This is the central carbon for the amino acid. This is called the alpha carbon. It is bonded to two functional groups, the carboxylate group and the amino group. It is also bonded to, this carbon is also bonded to a hydrogen, and in this case a methyl group. This is also called the R group, or the side chain. So amino acids differ because of this R group. There are different R groups present in different amino acids. So if we have to identify amino acids, we are going to look at this R group to identify the amino acid. In this molecule we are looking at, the R group is a methyl group. So again, an amino acid has a central alpha carbon, two functional groups, carboxylate and the ammonium group, a hydrogen attached to the central carbon, and a side chain or R group. So these are the different components that make up an amino acid. Now we have two groups of amino acids. These are the non-polar or the hydrophobic. Hydrophobic is water hating but not liking water. And polar, hydrophilic, is liking water. So how do we decide an amino acid is hydrophobic or hydrophilic, non-polar, polar? We are looking at the side chain. This is what determines a side chain. This is the carboxylate group, this is the ammonium group, this is the hydrogen, this is the alpha carbon, this is the R group side chain. We see that in this amino acid, the side chain is non-polar. Here the side chain is polar because of the presence of a carbonyl group. So that is the ID for an amino acid. If you want to call an amino acid a polar molecule, then it should have a polar component in this R side chain. So here the amino acid is valine. This is the abbreviation. There are two abbreviations, the single letter and three-letter abbreviations. So every amino acid, this amino acid has the abbreviation 3-letter, 1-letter. And this number we'll discuss in a little bit. When you are looking at every amino acid, you will have the complete name, the 3-letter abbreviation, the 1-letter abbreviation, and a value here. Okay? So now again, going back, amino acids are non-polar and polar based on the side chain. This is also in your book. An amino acid is non-polar when the R group is either hydrogen, alkyl, or aromatic. Here you are seeing glycine abbreviations. This is hydrogen, methyl. This is a hydrocarbon side chain, hydrocarbon side chain. All these side chains, aromatic side chain. So if you have any of these in the side chain, you are looking at a non-polar amino acid. Glycine, alanine, valine, leucine, isoleucine. So all these are non-polar amino acids. And every amino acid has a isoelectric point, pi. You call these pi, and we will discuss in a little bit what that number is. So these are the names and abbreviations and isoelectric points. So the numerical value that you see, 6.0, 5.9, these are the isoelectric points or pi points for the common amino acids. And this is at the physiological pH of 7.4. What does that mean? That this is within the body. The physiological pH of our body is 7.4. So if you are looking at a pH scale, the pH scale is 0 to 14. Midpoint 7 is neutral. Any number below 7, 6.9, 6.8, all the way to 1, below 1, those are acidic values. If you are going above 7, 7.4 is a weak base. So the physiological pH of our body is a weak base. So 7, 8, 9, 10, 11, 12, anything beyond 7, 7 is neutral. 8, 9, 10, 11, 12, 13, 14, all these are basic numbers. We are looking at a base. So you can see that at the physiological pH of 7.4, the 20 amino acids, not all of these are 20, but some of the 20 amino acid pi values, and these are non-polar. And because we do not see a polar group, we see aromatic, hydrocarbon chain, methyl, hydrogen in the side chain. Okay. Now, we are looking now at a group of polar amino acids. So amino acid becomes a polar group, a polar amino acid, when you have an alcohol, a thiol, or an amide in the R. So this is the R. Thiol is this. This is a thiol. We have talked about thiols. Amide, alcohol, phenol, we see aromatic alcohol. So when you have these groups in the R group of the amino acid, you are looking at polar amino acids. So these are the names: serine, tyrosine, cysteine, glutamine. You are seeing the name, the 3-letter abbreviation, the 1-letter abbreviation. And the entire table that we are looking at is on page 554. This is a good page to be on. If you are on this chapter, being on page 554 will be very important. You'll be getting all your information from this page. So remembering all the 20 amino acids, you look at your table. Okay? So how do you determine an amino acid is polar or non-polar? Is to look at the side chain. Again, we are reviewing the structure of an amino acid. Has an alpha carbon that is attached to 3 components: amino group, ammonium group, carboxylate group, a hydrogen group. 4th component is a R group that differs for each particular amino acid. A 3-letter or a 1-letter abbreviation derived from its name. So now we are going to learn how to draw an amino acid. Okay. Drawing an amino acid. So this is your carbon, alpha carbon, the carboxylate group, the ammonium group, hydrogen, and the R group. So aspartic acid. This is an amino acid, aspartic acid. This is its abbreviation, 3-letter, 1-letter, and this is the R group. This is asparagine. Again, this is the R group. This is the isoelectric pH, pH at physiological 7.4 pH. This is the pH, PI number. So this is how you set up an amino acid. Okay? Paying attention to the R group. And this pH is the PI number. Okay. Now, an amino acid is acidic when the R group is a carboxylic acid. So if the R group has a carboxylic group in it, the amino acid becomes acidic, 2.8, 3.2. Remember when we discussed a couple of minutes ago the pH scale, On the pH scale, 7 is neutral. So as we go below 7, the numbers get smaller and smaller. 2.8 is more acidic than 3.2. So as you are going down, it is getting more and more acidic. So the amino acids become acidic when they have a carboxylic group in the R. So you can see the pH values. You can look at the value and say that's getting more acidic. What are the two acidic? Aspartic and glutamic. And you can see the abbreviations. And we are going to remember the abbreviations. We are looking at page, which page are we on? 554. Okay? That's where all this information is. Now, amino acids are basic. Again, a basic solution has a pH above 7. Going to 14, that is the pH scale, about 7 to 14. So you are saying if you have an amine, ammonium, nitrogen and hydrogen, that bond is the nitrogen-hydrogen bond. If you have the amine in the side chain, here you have this group, here, your amino acid is basic. How do you know? The value is above 7. It's more and more. This is more basic. In all of these, this is the stronger. Arginine, lysine, histidine. These are basic. And how did we determine they're basic? We are looking at the side chain and looking at the presence of a NH bond, amine. Now we are going to determine these as polar or nonpolar. How do we determine polar or nonpolar? We are going to go to the alpha carbon. If this carbon is connected to a R group that is either hydrogen or a hydrocarbon chain, aromatic, you have a nonpolar. If you are looking at an alpha carbon connected to a R group with a hydroxyl, ethyl, carboxylic group, any of those, you are looking at a polar amino acid. So polar, nonpolar is based on this R group. Here the R group is a hydrogen. You see two hydrogens. So this is nonpolar and this is polar. We base this on the R group connected to carbon. This is hydrogen here, and this is a side chain with the OH group. That makes it polar. Draw the ionized form for each of the following amino acid and write the 3-letter, 1-letter abbreviation. So these are on page 554. Please refer, and you can see that we have set this up from page 554. So when you are setting up an amino acid, you have the central carbon, carboxylate group, ammonium group, hydrogen, and the R group. And then you have your 3-letter and 1-letter abbreviation for every amino acid. All amino acids except glycine are chiral. We have heard the term chiral before when we were in carbohydrates. What is a chiral carbon? And we have discussed this during our first lab too. A chiral carbon is a carbon with 4 different groups. And it has a non-superimposable mirror image, a chiral carbon. So this is your carbon. Remember Fischer projection, did we do that? Yes, we discussed Fischer projections. So we are setting, this is my chiral carbon. This is where the carbon is. L-glyceraldehyde has the OH to the left, D-glyceraldehyde is the OH to the right. These are the Fischer projections. I am setting up an amino acid, alanine. ammonium group to the left is L isomer, ammonium group to the right is a D isomer. So when we are looking at amino acids and we are going to decide the stereoisomers, we are going to look at the ammonium group like we have looked at the hydroxyl group for carbohydrates. We are looking for ammonium group in amino acids. So again, L-cysteine, say amino group is to the left. D-cysteine amino group to the right. So all amino acids except for glycine. So I want you to look on page 554 and pick out the molecule of glycine, and I'm doing that myself. I'm looking at glycine is the first molecule that starts off the page 554. I'm looking at the carbon, the central carbon. I see 2 hydrogens attached to the carbon, 2 hydrogens. So we do not have 4 different groups, and that is the reason glycine is the only achiral carbon. So if you do not have 4 different groups, the carbon becomes achiral. It has 2 hydrogens, 2 of the same thing. D or L isomer, so enantiomer, stereoisomer are also called enantiomers. So I'm looking at the ammonium group to the left, ammonium group to the right, and this decides D or L. Ammonium group to the left is L, ammonium group to the right is D, DL. Now link to health, important component. Out of the 20 amino acids used to build proteins in the body, 11 can be synthesized in the body. 9 are essential amino acids. We call them essential because they are essential part of our diet and have to be obtained from the protein in the diet. So the essential amino acids are listed here, the 9 acids. Amino acids are listed, and these have to be a part of the diet, and therefore we call them the essential amino acid. Okay, so eggs, milk, meat, and fish contain all essential amino acids. Of all the 9 are present in milk, eggs, meat, and fish. The incomplete number of amino acids, not all 9 are present in grains, beans, nuts, have amino acids, but not all 9 of them. They are deficient in 1 or 2. And you have this information also available in the unit with every individual component listed with the deficient amino acid.